Okay, the next topic we'll be covering is protein folding. And before we get into protein folding, there's a couple of things that we have to discuss to make this topic a little bit easier to understand. Okay, so the first thing we're going to review, a topic we discussed a while back, is that in an aqueous solution, water will minimize its interaction with hydrophobic groups or hydrophobic surfaces. I like surfaces better as a word to use because that encompasses whether the whole molecule is hydrophobic or part of a molecule is hydrophobic. That's why I use the word hydrophobic surfaces. The words hydrophobic surfaces. Okay, so what does that mean? That means that when H2O is mixed with hydrophobic surfaces, that it will try to minimize. H2O will try to minimize its interactions to almost zero. It will try to get to 0%, where there's 0% contact between H2O and the hydrophobic surface. So we'll just go for that maximum amount of separation. So 0% interaction between water and hydrophobic surfaces. Now, this process of going from H2O mixed with hydrophobic surfaces to 0% interaction between H2O and hydrophobic surfaces has a delta G that is less than 0. So it's a spontaneous process that will happen by itself. This is a favorable event, meaning that entropy is decreasing. So entropy here is higher than entropy on this side. So the left side is actually more ordered than the right side. Okay? So that's one thing to keep track of before we start talking about protein folding. So what's the second thing? So we said that if you have a positive charge and a negative charge, that this is a preferred state where these two things are electrostatically interacting with each other. The process of separating them from each other, where they're no longer interacting, those three red dots are not between them anymore, this is unfavorable, meaning this actually has a delta G greater than 0. Right? So you need an input of energy to separate charges from each other. Charges would like to pair with the opposite charge. So a partial positive charge would like to pair with some sort of negative charge, with whether a negative or a partial negative, etc. You can think of all the different combinations. That's really not important. What is important that a charge will like to pair with its opposite charge. So therefore, overall, you have neutrality. So let's look at a polypeptide chain. We'll draw a short polypeptide chain. And of course, you have side chains. Let's label our side chains like this because we know that there are really two types of side chains, right? Let's scroll up a little bit. So we have nonpolar, and we'll just put nonpolar as red. And we have polar. Of course, within polar, there's polar uncharged and polar charged, right? But we're just going to separate them into two categories because the hydrophobicity and hydrophilicity is what's important to us. Because the polar would be hydrophilic and the nonpolar would be hydrophobic, right? So let's say that this chain, I have a hydrophobic here and a hydrophilic here. And let's say that I have another hydrophilic, then I have a hydrophobic, and another hydrophilic. So that's our distribution of amino acids, our sequence from the N-terminus to the C-terminus. We're not really interested in the actual identity of the amino acids. We just want to know if it's a polar or non-polar amino acid. So I'm going to erase this right now. We know that non-polar is red and blue is polar. So the polar side chains are hydrophilic. They have some sort of charge on them. That was our definition of hydrophilic, meaning that if you have a hydrophilic molecule, you're going to have some sort of partial positive, partial negative, or a full negative or a full positive on them, right? So those charges will have to interact with some sort of opposite charge. Well, that's fine because you have H2O molecules that can interact with it, right? You're in an aqueous solution, there is H2O molecules. Also, there are other hydrophilic molecules in the solution. So these polar side chains can easily interact with any hydrophilic molecule in the solution, including water. So we'll just draw a couple of water molecules here. And then they're interacting with these things. So these side chains are happy. What else is hydrophilic here on this polypeptide chain? Well, we know that this oxygen right here has a partial negative on it, right? So does this oxygen. So does this oxygen. So does this oxygen. So does this oxygen. And of course, you see the big negative charge on the carboxylic end. So these all need to interact with water also. So what we're going to do, instead of just writing out water molecules, we're just going to sort of circle these things green. So I'm going to remove the water molecules. So this needs to interact with water or hydrophilic molecules in solution. This needs to interact with water or hydrophilic molecules in solution. So there's this. So there's this. So there's this. 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 And this, right? Okay, what else? Well, we know that the hydrogen that is bonded to the nitrogen and the amide bond or the peptide bond is partially positively charged. So that will also need to interact with water. So let's go ahead and color these blue. And of course the NH3 plus, that's a big one too. So I'm just going to color the whole thing blue for that one. So this right here, this right here, this, and this. All of the things that are circled in green need to interact with water or another hydrophilic molecule that's in the aqueous solution. Or they have to be paired with an opposite charge somewhere. Because if they're not, that's a high energy state as it's being shown here, right? So we have a bit of a problem. What's the problem? Well, these R groups that are red, these hydrophobic R groups, they need to be excluded from solution. So let's circle these red. Well, how do you exclude these hydrophobic side chains from solution? Well, all the things that are circled green are able to still hydrogen bond or ionic bond with an opposite charge through some sort of electrostatic interaction, right? How do you accomplish that? And that is the problem that protein folding solves. So let's go ahead and take a look at the topic. How does a polymer of amino acids fold into a three-dimensional structure, a unique conformation which has biological activity? So exclusion of hydrophobic side chains from solution. So here we have a polypeptide chain right here. And the gray line is the polypeptide backbone. And all the little color circles are the side chains that are sticking off of the polypeptide backbone. Now, the nonpolar side chains in this picture are actually green. Nonpolar is green. And all the other ones are polar. So you can see that once this polypeptide chain in its completely unfolded state meets the aqueous solution, what will happen is, initially, the hydrophobic side chains are excluded from solution. And that's what's happening in this picture. You have this hydrophobic core region which contains the non-polar side chains, while the polar side chains are sticking out on the surface of the protein facing solution. This will happen very quickly. It's called the hydrophobic collapse, where the hydrophobic side chains are excluded from solution and put into the core of the protein away from the aqueous solution. So the aqueous solution is no longer interacting with those hydrophobic surfaces. But the problem is this. Since these hydrophobic surfaces are covalently attached to the polypeptide backbone, they also carry regions of the polypeptide backbone into the hydrophobic core. Like this region right here, and here, for example, and here. These are all stuck in the hydrophobic core away from solution. But what happens to those amide hydrogens and those carbonyl oxygens of the polypeptide backbone? These right here. Right? What happens to these guys if they are shoved in to the hydrophobic core of the protein away from solution? The hydrogens have a partial positive, the oxygens have a partial negative charge on them, and they need to find a partner to interact with each other. So, kind of the answer just came out, right? I just kind of said it, gave you guys a little bit of a hint. What was the hint? Let's remove these circles real quick. Well, the hint was that the oxygens have a partial negative charge on it, and the hydrogens have a partial positive. And they are in the hydrophobic core away from any other molecule or anything that they can hydrogen bond with. Right? But they're near each other. And they're opposite charges. One is partially negative, one is partially positive. So formation of secondary structures, alpha helices and beta sheaths. The polypeptide backbone is hydrophilic. During stage A of protein folding, parts of the polypeptide backbone are dragged into the hydrophobic core and are away from solution. When the polypeptide backbone is in solution, it can hydrogen bond with water or other hydrophilic molecules, as we said before. When away from solution, bit of a typo here, when away from solution, the only hydrogen bonding partner is itself. What we just said. Alpha helices and beta sheets are formed in order for the following to occur. Hydrophobic side chains are excluded from solution, and the amide hydrogen, NH, and carbonyl group, CO, of the polypeptide backbone have hydrogen bonding partners within themselves. So, if we go back and look at alpha helices and beta sheets that we talked about previously, right? Here's our beta sheet. Well, the beta strands, the polypeptide backbone of the beta strands, are hydrogen bonding with each other. And in the case of the alpha helices, a hydrogen bond is formed between every fourth peptide bond between the carbonyl oxygen and the amide hydrogen. So you have hydrogen bonding interactions occurring between the polypeptide backbone when an alpha helice is formed, and also when a beta sheet is formed. So the formation of these secondary structures is to solve that problem. The hydrophobic surfaces need to be excluded from solution. But the polypeptide backbone is being dragged in to this hydrophobic core of the protein where there is no aqueous solution. There is no water or any hydrophilic molecule for them to hydrogen bond with or interact with electrostatically. So, To solve that problem, secondary structures are formed, alpha helices and beta sheets, where the polypeptide backbone is hydrogen bonding with itself. Therefore, the process of protein folding is an attempt by the unfolded polypeptide to find a conformation. And just a quick word, conformation just means the three-dimensional structure, where all the hydrophobic surfaces are excluded from solution and all charged surfaces, partial or full, have a partner of opposing charge to interact with. So initially what you have is the hydrophobic collapse. The hydrophobic side chains are excluded from solution. Next, what will happen is the polypeptide backbone will start to wiggle around in that conformation, trying to find a way to hydrogen bond with itself, and you get the formation of beta sheets and alpha helices. Stabilization of the native structure of a polypeptide. The final tertiary structure of a protein, the polypeptide, its folded state is stabilized by many weak interactions. And remember we said, when you have many weak interactions, that is strong or stronger than covalent bonds. And those include all the ones that we learned about. Electrostatic interactions between full negative and full positive charge, for example, acidic and basic amino acid side chains, that is being shown here and here. Hydrogen bonding, which is being shown here and here. In this case, for this one right here, this is between the backbone. And van der Waals interactions, when all those hydrophobic side chains are moved to the hydrophobic core of the protein, when the protein collapses in its folding pattern, all those hydrophobic surfaces are brought near each other and van der Waals interactions start to occur, and you have a bunch of weak interactions in the hydrophobic core also. The many weak interactions that form lead to a stable state that can be only disturbed if sufficient heat or chemical denaturant is applied. So denaturation is a process where a protein has lost its native structure and no longer has the activity level it once had, either full loss of biological activity or partial loss. And you can kind of see this in processes like cooking. If you take a piece of meat, like let's say a steak, before you cook it, it feels different, right? And once you cook it, it has a different type of feel to it. The texture of the meat changes. And that's because the process of cooking will denature those proteins. And now they're in a structure that is very different than the structure that they had before. In the case of some proteins, when the denaturing conditions are removed, the protein refolds into its native structure. So there are some proteins that when they're denatured, for example by heat, you heat them up, they unfold, you cool the temperature down back to normal, and they refold back to their native structure. In other cases, when a protein is denatured, it will not be able to fold back into its native structure. In the absence of denaturant, it will fold into a conformation which lacks biological activity. Cooking foods, for example. If I cook a steak, just because I let the steak cool down or I put it in the fridge, I don't go back to the state that was the state of the meat being uncooked. Many proteins, when they're denatured by heat or chemical denaturant, when you remove that denaturing condition, they will actually fold into a non-native structure that has no biological activity. And something else has to happen for them to refold back to their native structure. And typically that something does not occur in the cell. At that point, if you have a denatured protein in a cell, the cell will simply remove that protein from circulation. It will break it down to the amino acids so that a new protein can be made from transcription translation. Many proteins, such as enzymes, have a narrow temperature and pH range in which they have 100% activity. Being slightly out of those optimal conditions in either direction, they will start to lose activity very quickly, but will regain activity when the conditions return to optimal. This is not denaturation, as the protein does not unfold, but rather has a very small change to its conformation, which in itself is enough to decrease the biological activity. So if you're just kind of going a little bit to the right and the left of the optimal conditions, and you lose a little bit of activity, or a lot, maybe you lose 100% activity of that protein, whether it's an enzyme or some other protein with some sort of biological activity within the cell, but you regain it when you come back to optimal conditions. That is not necessarily unfolding. Unfolding is an event that the cell will avoid at all cost. Okay, so we'll stop this video here and continue later. Bye-bye.